Hello, everybody. Really great to be here today. My name is Professor Will Srubar. I'm an associate professor of civil, environmental, and architectural engineering and affiliated with the material science and engineering program at the University of Colorado here in Boulder. Really excited to be here today to talk a little bit about our work related to carbon storing bioarchitecture. And really what I want you to walk away with is an understanding of what we're really trying to do. We're trying to really genetically program single cells to help us build materials and structures of the future. So I think it'll be a pretty exciting time today. So as I mentioned, today I am an associate professor, but that was, of course, not always the case. I'd really like you I'd really like to take you back to the beginning so that you can understand really my worldview and how I view the current world and really the future. So I grew up on a cattle ranch and a cotton farm on the plains of Southeast Texas. And really, growing up, the land was our livelihood. And so every day I really came face to face with what I call the cleverness of nature. You know, nature has figured out a lot of really cool things, and they respond very, very well, especially in times of crisis. For example, I really remember walking out right after a flash flood and seeing piles of ants that have piled on top of each other, really to ensure their survival. And I remember, you know, as a little kid, chasing around lizards and delighting when they would lose part of their tail that continued to wriggle to evade me, which was the predator. So I was always very intrigued and surrounded by nature and intrigued by its responsiveness, especially in times of acute crisis. And while I had a very rural upbringing in Texas, I wasn't necessarily completely isolated. So we were very close to Houston, within about an hour. And it really was the skyline of 1980s and 1990s Houston that inspired me to become a structural engineer. And I remember going to baseball games with my dad at the Houston Astrodome and just sitting there and marveling at the abilities of humans, men and women, our ability to design such large-scale structures that have such a profound effect on humans and the built environment as a whole. This really came full circle for me. My very first project out of undergrad was to work on the Dallas Cowboys Stadium in Arlington. So I really was lucky to have that experience working on such a landmark project that reminded me so much of why I got into structural engineering to begin with. So as I got a little bit older, you know, what I realized was I was the generation of my family to urbanize. And I moved into concrete jungles where it was very apparent to me that humans were so separate from nature. We have these really stark images and examples of Central Park, for example, where we know what is human and we know what is nature. And you know, I was very infatuated with the materials with which we choose to build, the materiality of our urban environments, but also how it's very much abiotic, not alive. But you cross the street, for example, in Central Park or other park and recreational spaces in cities, and we don't have the abiotics. It's very much alive. And so as I progressed in my professional career, I really toyed with the intersection of natural and built environments. And I began to really question convention. So a lot of my research at the University of Colorado asks these questions, like, why are humans so separate from nature? especially our built environments. Why do living things grow and regenerate and respond and adapt, but our buildings, once they're built, generally do not? And really, what can we learn from our fellow species? Like I said before, I do believe that nature has figured out a number of different problems. We just have to know really where to look. So at the University of Colorado Boulder, like I said, I'm a professor. I teach courses, but I also lead the Living Materials Laboratory, which is a materials research laboratory. And I'm very fortunate to work with a number of very talented students and postdoctoral scholars, where we try to engineer biomimetic and living materials for the built environment. And as you can imagine, this requires disciplines and expertise, not only in civil engineering and architecture, but in material science, in biology, in genetics, microbiology. And so our day-to-day is very exciting. It's learning how to communicate, it's learning how to solve problems together. And a lot of what I'm going to show you today really is a tribute to the strength of the interdisciplinarity of the team and the talents that they bring to the table. So we work on a number of different material systems, and as I mentioned, we really try to play with the interface of what is human and what is nature. And in a nutshell, we're really trying to bring building materials to life. And I don't really have time to go into all of these different material systems, but in general, what we try to do is take biological materials, more than just wood, take other biological materials like proteins or sugars or lichen, and use those in engineering applications specifically for the built environment. Or we use living organisms themselves. Lichen is an organism comprised of fungi and cyanobacteria that live very symbiotically, that can impart some very, very cool advantages to indoor environments like CO2 sequestration and moisture buffering, all the while serving as an ecological toxicity indicator. It will change colors if there's something harmful in the air. One project I will talk about is our work with engineered living materials and using photosynthetic organisms to make a biocement in lieu of ordinary Portland cement for, again, making structural materials specifically for the built environment. But really where I wanted to start is really a conversation about the current landscape of biomaterials for building and construction. And usually when I start to have these conversations with folks who are curious to learn more about biomaterials, the conversation usually starts in the forest because we have been using biomaterials in the form of wood and engineered wood products for centuries and really millennia. And if you think about it from a fundamental standpoint, nature grows this magnificent structural building material from a genetically encoded single seed. So the genetics, the morphology of the tree, how tall it will grow, what it will look like is all genetically encoded in the DNA of a seed. And we recognize the power and importance of that and really the terrific material properties that this process creates with trees. But we recognize too that this process can take years and even decades to get a mature tree. So, a lot of our work toys with how can we speed this up? Maybe we need to move away from woods and wood-based materials to more rapidly grown materials, especially those that are carbon storing. Because we all recall that trees are photosynthetic, they absorb CO2 to help build the biomass. And what's really unique about trees and really any other biomass you can think of is that trees, plants, grasses, straw, bamboo, even algae, if you take the dry weight of those materials, of that biomass, 40 to 50% of it is stored carbon dioxide. which is certainly one of the main advantages for using some of these materials in our buildings. Because if we take a look at the CO2 emissions of conventional construction materials, so we're talking about steel or glass or concrete, and we look at the overall life cycle of that material and the CO2 emissions that are associated with the material production, use, and disposal, you can see that for conventional materials, we have nothing but emissions. So take steel, for example, where we mine the ore, we process it, we go through the steel making process, we transport it to the construction site, and there it sits for many years. And certainly after its lifespan, we can recycle it. Steel's really great in that we can recycle steel. But the main point I want to draw with this particular part of the graph is that no matter what, we still have positive emissions, meaning that even if we try to reduce this as much as possible, we still will have emissions associated with the manufacture of the materials. Now, if we take a look instead at photosynthetic materials, biomaterials that utilize CO2 in the growth of the biomass, we can see that yes, there are some emissions associated with planting, the initial planting and cultivation of the plants or biomass, but all the while the plant or algae is growing, there's this photosynthetic drawdown. So as the tree grows or as the cell divides, we certainly have more and more photosynthetic drawdown of CO2. And so then if we take that biomass and then go through the subsequent steps of making a material, transporting it to the construction site, and then storing it in buildings, what we have as an advantage is meaningful CO2 storage in our buildings instead of carbon emissions, that embodied carbon that we would otherwise see with other materials with which we choose to build. And certainly the choice at the end of the building lifespan will determine net carbon emissions post-use. But all this to say that utilizing photosynthetic materials in our buildings is far more advantageous than other materials. And we recognize that climate goals to meet the Paris Climate Agreement and the goals of not exceeding 1.5 degrees Celsius rise in global temperatures, we must not only focus on reducing the carbon emissions associated with materials like concrete, steel, glass, aluminum, other metals, because we will continue to use those materials, but we really need to focus on the sequestration and storage and balancing those emissions with the storage of carbon in photosynthetic materials to get at carbon neutral and even carbon storing buildings. So, you know, many of us know this, and folks in Canada and especially the Pacific Northwest in the US are already well aware that timber and engineered wood is a good strategy for reducing our embodied carbon in buildings. And we know that engineered wood is in our future with the onset of CLT and mass timber. And so that's not necessarily going away. But what I want to focus on more in this talk are the other photosynthetic materials that exhibit this photosynthetic drawdown more rapidly than growing trees in a forest are really the key to getting us closer to carbon neutral and carbon storing buildings rather than relying solely on wood products. There was this really great article in Nature Sustainability by the Yale School of Forestry that really did show that it is possible, based on some assumptions that have to do with global supply of forests, that we could get much closer to transforming our buildings from carbon emitters into carbon sinks. But what I really do think the article was a little bit short-sighted in highlighting was the ability for other rapidly grown photosynthetic biomass, just like straw or bamboo or hemp, cork, other natural fibers that can be used to reinforce rammed earth. Those types of materials are going to play, in my opinion, a key role in transforming the built environment from a carbon emitter into a carbon sink. So I'll highlight some examples that many folks are probably very well aware of. Ecovative's mycelium foam is a good example. This is a grown fungal mycelium product where agricultural biomass, agricultural residue is used as a fiber reinforcement, and a fungus is used to grow as the binder, kind of grow around the particles to bind them together. And what results is a material that exhibits many similar properties to conventional styrofoam and other thermal insulation. And many of you may recall that there was an installation at the MoMA in New York City, demonstrating the ability to manufacture these materials at scale and to use them in building type applications. One of the challenges that I know Ecovative has faced over the years is cost parity with many other types of insulation, without a lack of drivers for designers and decision makers to choose this material. And so what Ecovative has really found is more application of their materials in more higher end fixtures and finishes. And there are other examples that like this, where agricultural biomass that's rapidly grown can be transformed into a higher value product. One example is hempcrete, which I do believe the Canadian community is much more familiar with hempcrete than my US colleagues and counterparts. But for those of you who don't know, hempcrete is a highly insulative, non-combustible material made out of the woody part of a hemp plant that grows extremely fast, and that is combined with a lime-based binder that slowly carbonates over time. And it's used widely today throughout parts of Europe and other parts of the world, and I know in Canada, but it really has not yet found many applications here in the United States. One exciting thing that my research group found was that hempcrete, if you do the full life cycle assessment of hempcrete can be a carbon negative material, meaning if we make more of hempcrete, we will be pulling and storing more CO2 out of the atmosphere the more of those materials that we make. But it really is mixture dependent because many hempcrete formulations do add a little bit of Portland cement. Many of you know Portland cement is certainly a hotspot in concrete for carbon emissions. But what you're looking at here are examples of life cycle assessments for 8 different mixtures of hempcrete. And what we're showing is that based on the mixture proportions, you do get various emissions, carbon emissions associated not only with planting the hemp and producing the hemp, which is the dark green, but also the dark purple is really the emissions associated with the binder. So the lime and the Portland cement, if it is used. What we do show then on the right-hand side of each of these graphs for each mixture is the potential storage, carbon storage of the mixture formulations, both due to carbonation of the lime-based binder, but really the biogenic carbon storage of the hemp shiv. And you can see that because many of these graphs dip below the zero, y equals 0 axis here, you can clearly see that some mixes, some better than most, or some more than most, and even some may not get there at all. but there are certain formulations of hempcrete that account for all of the emissions that were associated with its manufacture, but also if you account for that biogenic carbon storage, that you do end up with a carbon negative material that's fire resistant, moisture resistant, mold resistant, et cetera. So we were really excited about this. There have been some other LCAs of hempcrete in the literature, but to our knowledge, this was really the most thorough LCA that was performed. This is now published in the Journal of Cleaner Production if you have interest in learning a little bit more about it. But what we did was I had a very interested PhD student who is helping spin out some of this knowledge and this technology into a company. So Minus Materials has spun up as a company out of my lab that's really focused on manufacturing only carbon negative building materials such as hempcrete. And we're really positioned well here in Colorado in the United States because we are arguably leading the country with hemp production, and other states are soon to follow. So if you look around, for you architects and engineers who are looking for carbon negative solutions, there are a myriad of other carbon storing materials from plants, and a lot of them are derived from wood and timber, forest products. But there are many examples, such as those shown here, that look at other types of rapidly grown biomass, like straw board. Rewall looks at recycled cardboard. Eco Cocoon looks at even straw, cellulose, timber, composites, and AgriBoard, which also uses agricultural biomass in the production of their materials. And so I certainly encourage everyone to look at these materials, ask for EPDs, because many of these products don't yet have EPDs. But when you are doing your whole building life cycle assessment, it is important to pay attention to shipping and logistics, because what we're finding is that many times a lot of these materials from agricultural residues must remain regional for them to be cost competitive. And so if you actually take that into account in your life cycle assessment as well, these will start to be a little bit more attractive from a whole carbon standpoint. Some materials too are sourceable from Europe, but again, it's really important to pay attention to transportation, because that can come into effect. So the rest of my presentation, I really want to highlight some results from our research coming out of my lab, really that has spun out of us really asking a provocative question as to whether or not we could program bacteria to grow carbon negative building materials, and therefore, logically extending that to structures and even buildings, because we thought that there could be a better way than to rely on seasonal growth of biomass, such as straw, hemp, and other grasses, or bamboo, or even the decades-long cultivation of trees. One of the benefits or there are a few benefits of turning to microorganisms to help us manufacture materials. Number one is time. So many of you remember your second grade and third grade science classes where you learned that bacteria have exponential growth rates. So if we allow these bacteria to grow unencumbered, we can get exponential growth versus say linear growth that we may otherwise see with other plant-based approaches. Number two is soil and land resources. We don't necessarily need microbes to take up any land or soil. Number three, it's bacteria autonomously growing, so they can do their thing, they can self-regulate, they can even communicate with other cells. And then number four, specific bacteria, which is what we were very interested in, are in fact carbon storing. And what we're talking about are photosynthetic microorganisms. So this is a very zoomed in image of bacteria, cyanobacteria. Again, science, everybody kind of knows cyanobacteria are kind of Frankenstein green because they are photosynthetic, just like plants. So what they utilize is CO2 and sunlight as really the main sources of energy, and they grow and replicate, again, at exponential scales by producing more and more biomass as part of their cell walls. And then many of them have unique capabilities to make materials. And so we've been paying more attention to how these photosynthetic microorganisms can be leveraged to help us grow certain materials that may be useful for buildings. So what the cultures look like in the lab is kind of what you would expect to see. You know, the greener, the deeper green the media is, the more biomass and the more cells there are in the media. It's very easy for us to tell whether or not the bacteria are alive and happy or not. It's whether or not they are green like this or not. So like I said before, we feed these bacteria light, carbon dioxide, which is what they need, and a little bit of nutrients, a little salt. You know, they grow really well in an artificial seawater of sorts, and they grow and replicate and again build biomass in the process. What is particularly exciting about this is that, you know, while we're operating on the lab scale, studying these microorganisms, we know that there is large scale cultivation of algae that is ongoing today. So a lot of you might be familiar with algae cultivation for biofuels. So a similar concept where we have these very large scale cultivation ponds where algae is grown, the biomass is captured, and then it is burned for energy and heat. So that's kind of a carbon neutral approach to creating heat and energy. But algae are also cultivated at this scale for other useful purposes, such as pharmaceuticals, biofuel, like I said, food, fertilizer. You can even buy algae pellets as a supplement from natural food suppliers. But what we really wanted to do is utilize algae, like cyanobacteria and other microalgae, to make building materials. And we just had to know at which organisms to look and where in nature we should look for inspiration. So what was particularly captivating for us was that there is a specific group of cyanobacteria, marine cyanobacteria that live in the ocean, that grow strong and tough minerals. So on their cell surface, in the right environments, they will precipitate a mineral, and that mineral, under the right conditions, can grow into a larger structure. This is an example of stromatolite formation. So stromatolites are the mineral that is a calcium carbonate-based mineral that is formed by some marine algae, so cyanobacteria that live in the ocean, and that over time nucleate crystal on crystal on crystal to really grow these rocky calcium carbonate formations. So there's really strong, tough mineral formations that really lends itself well to a structural building material like a concrete alternative. So we were really inspired by this process of stromatolite formation, really understanding biomineralization by these carbon storing photosynthetic organisms. And we wanted to grow, for the lack of a better word, grow a concrete alternative in the laboratory. And we focus on concrete a lot. And I just want to remind everyone, you know, why is concrete such a carbon hotspot? So, many of you are very familiar with concrete, where it's kind of like baking a cake. We combine Portland cement, which is like the flour. Cement is to concrete as flour is to cake. We combine the powder with water, and we mix coarse and fine aggregate together, as well as supplementary cementitious materials that are largely rich in silicon. We replace some of the Portland cement with these supplementary cementitious materials like fly ash and slag and metakaolin and other natural pozzolans, to do two things. One, to replace Portland cement, because Portland cement is the most carbon and cost intensive constituent, but also number two, that the chemistry works better. So what results is a concrete that is actually more strong and more durable than if we were to just use Portland cement by itself. And then we use a number of different secret sauces, chemical admixtures to tailor and tweak both the fresh and the hardened state properties of concrete, usually in small doses. But really, concrete is such a carbon hotspot because of Portland cement. And I want to walk through this process because it's very important to understand that if we continue to use Portland cement how we create it today, we will not ever get to a carbon neutral Portland cement production. And it's primarily because when we start at the very beginning of Portland cement production, we take limestone and clay, which has iron, silicon, and aluminum. We put limestone and clay together, we crush it, and then we start heating it up. And once we start heating calcium carbonate, which is limestone, once we start heating the limestone up, it undergoes a chemical reaction called calcination. And off goes all of the wonderfully stored carbon that was in our calcium carbonate. So this process of burning limestone accounts for about 50% of carbon emissions associated with Portland cement production. The other 40% comes from fuel combustion and another 10% from transportation of the final product to ready mix plants. And so what I like to tell audiences is that we can reduce the emissions associated with combustion and transportation if we switch to renewables and find alternate sources of energy. What we cannot do is get away from this calcination of the limestone unless we have some carbon capture and storage mechanism on every single cement plant in every part of the world, which is largely infeasible. So really the principal carbon reduction strategy for concrete has been to use less, or in our case, no Portland cement. So going back to our stromatolites, using stromatolites as inspiration, we grew a structural material in the laboratory using these biomineralizing photosynthetic cyanobacteria, only using sunlight, CO2, and artificial seawater. And as an added bonus, we actually kept it alive and kept the bacteria alive inside of it. How did we do this? So we took all of what we would call the abiotics, so the off-the-shelf additives, the salts that are needed to replicate kind of an artificial seawater that the bacteria would need. We did add a little bit of gelatin for the bacteria for a couple of different reasons, partly to retain water, but also as a proteinaceous material. Gelatin is actually really strong, and for centuries has been used, some form of collagen or gelatin has been used for centuries as adhesives and really bindings of wood products. So we added these together with our photosynthetic bacteria. We exposed the bacteria to sunlight and CO2. And what we ended up with was a living building material. And I'll show you some photos of it here in a second. But what was great is that we could control the moisture and the temperature of the material so that we didn't harm the bacteria in the making or the manufacturing of the original, what we would call a parent brick. What that enabled us to do was split the parent brick in half, add more abiotics and more media, and allow and sand and allow the bacteria to grow into the second half of the material, so that one parent generation became two in the first child generations, and we did that 2 subsequent times. So one parent generation actually resulted in the growth of 8 child bricks of the same dimension. And we envision utilizing these materials for a certain amount of time, but then these materials really lend themselves well with post-use recycling, where we can just take the material and kind of recycle it back into the original manufacturing process. So here's a lot of images. I'm going to walk you through exactly what I'm showing you here, starting along the bottom left. So like I said, we started with the culture of sand and media and cells. If we zoomed into what was going on inside, so these green spheres are our sand particles, and the tiny dots are bacteria, and then the gelatin proteins are kind of these green lines. But as this material is growing and as the bacteria are growing and growing more biomass, they're also precipitating calcium carbonate. So they're taking in the CO2 and not only growing and replicating their own biomass, but they are also storing more carbon dioxide in the form of a mineral. And what that enables is an added toughness and an added mechanical property to help bind those sand particles together. And so once we cool the material down, because bacteria like to grow at a little bit higher temperatures, once we cool it down, what we end up with in the upper right here is kind of an ooey gooey brick that's filled with bacteria and these precipitated little biominerals. And if we dry out the material by decreasing the humidity, we get a material that exhibits properties very akin to cementitious mortar material, kind of like the mortar that's used in between your bricks in your house. And you can imagine that interrogating this trade-off of high humidity, low humidity will affect mechanical properties, and it will also affect regeneration ability, because bacteria like ooey gooey environments, whereas structural engineers like drier environments because you get more mechanical benefit. So our seminal paper was published this year in a journal called Matter, and it really was just demonstrating that we could use this approach to grow structural materials without the use of Portland cement, but utilize that stromatolite, tiny stromatolite formation inside of the brick to help create a biocement to help bind the sand particles together. But if you look at this more broadly, different, we certainly aren't married to gelatin. We could use different hydrogels or different bacteria inside of the system, could enable a living building material with a multitude of different functions. And what we really were excited about as well is that we proved that we can make materials at any scale. So this is a 600 centimeter cubed brick. DARPA, the U.S. Department of Defense, funded this work, so it was a little bit of a marketing scheme here, but large scale samples, and we are able to, as I mentioned, take one parent brick, and after 3 subsequent growing, we could grow a generation that is 8 bricks from one parent generation. We also showed that we can form arbitrary shapes, and we can move away from just what we would call living materials to living structures. So all of you structural engineers out there know that efficiency comes not only with material properties, but also the shape and form that it takes. So we're able to make very complex shapes pretty easy because we're able to form the material. And so this is also just an example of how we are moving away from just making a living material really to a living a living structural element. This is through a program that performs topology optimization. It really is a form-finding software that shows you the most efficient shape for the structure given the loads and given the material properties. So it's just our first go at demonstrating that we'd be able to do this at a scale that is tangible and touchable. This is also a technology that's spinning out of my lab. So there's another startup effort really looking at a biocement that's based on this technology. And what we're doing is that we're taking the cyanobacteria that we use and we're spray drying it in much the same way as you would spray dry yeast and have a yeast packet for your sourdough culture. We're doing the exact same thing for a starter culture for a grown biocemented brick. So we're taking the engineered cyanobacteria with our nutrients and packaging that all together to where you could have a just add water biocement and grow your bricks on site wherever you are in the world. So all that would be needed to form a brick would just be to add water, expose the material to sunlight and CO2, add any type of scaffold that you would like. So we use sand, but you could use other agricultural waste or other types of waste like recycled glass or plastic, and result in a grown biologically active material that exhibits mechanical properties just like cementitious mortar. And I will say, you know, photosynthetic microorganisms are very innocuous. They will not kill anyone. It's the same type of bacteria that's growing on your fish tank. It's very, very akin to algae, so it isn't harmful to humans or animals. You know, we're particularly excited about this as well, not only for terrestrial applications, but if you kind of think about you know, humanity's goals for the next few decades, this could be a technology that is brought to the moon or even to Mars, where there are very limited resources to build infrastructure there on the moon and other planets. What we do have, though, in those environments is a lot of sunlight. We do have access to water, at least on Mars, and we certainly have access to CO2 as well on Mars. So you could think that it's not too far-fetched to think that a technology like this, we would be bringing it to extraterrestrial, to build our extraterrestrial developments, because I don't think that we'll be hauling steel beams or bags of cement with us when we go to these places. So we've been pretty fortunate to get a lot of really good press lately. We were in the New York Times and have gotten a lot of great feedback on our approach, especially from a carbon neutral and carbon negative standpoint, as it pertains to growing a concrete alternative. And you know, like I said, we're very interested in advancing this technology, looking at other substrates, looking at other biological functionalities that we could impart theoretically to building materials, and really recognizing that bacteria, even cyanobacteria, we have the ability to genetically program their functions in much the same way that we would genetically program other bacteria to make useful fuels or chemicals like or pharmaceuticals like insulin. Most insulin is actually produced by engineered bacteria, saves a lot of lives. So too do we think that we can boost the performance of these bacteria to make more useful materials for us. So this really speaks to this idea of genetically programmable architecture, genetically programmable material manufacture, both at a small scale, but then hierarchical organization into larger structures, all autonomously programmed into the bacteria, where we humans don't have much need for you know, input into that process. And we're already starting to address this. This is the last project that I'll highlight that's coming out of our lab is we recognize that if we tailor the genetics of the cyanobacteria, we can actually get them to grow different shapes and morphologies of minerals. And by tailoring not only the genetics, but also the conditions in which it's grown, so changing the temperature, the light levels, and other physical, I'd say, effects of the bacteria during growth, we can get these magnificent morphological, really a diverse set of biominerals that are carbon storing. So this is limestone that has been produced by the bacteria that take carbon dioxide from the air and fix it in a limestone mineral. And what we're really trying to do is grow aggregates in concrete. So there are a couple of companies out there that are also trying to make aggregates from carbon dioxide. Our approach is purely biological, whereas others take a look at physical or chemical processes. And why this is so attractive is that if we take a look at a cubic meter of concrete, 90%, about 90% of the volume of concrete is aggregates. It's the sand, it's the coarse aggregate. 95% of the carbon emissions actually come from the Portland cement. So if you think about limestone aggregates, there is a tremendous opportunity to store carbon dioxide in the form of newly created calcium carbonate limestone aggregates in our concrete. So there is the possibility to create a carbon negative concrete if we pay attention to the aggregates and how we manufacture them. Again, we're looking at biological approaches to manufacturing. And just to put this into perspective, if we were to grow our materials, our limestone particles, and use that as a filler to replace 5% of the Portland cement, what that would actually equate with is a 10% carbon reduction, because we're not only replacing the Portland cement, but we're doing so with a carbon negative material. So this is just a quick example of where we're really heading. And if you'd like to know a little bit more about this, certainly reach out. We're happy to keep you up to date on our work and even our efforts at trying to spin this technology out of the lab as well. We're not the only folks who are taking a look at photosynthetic organisms. There are certainly other folks looking at algae and how algae can be grown in buildings or used in buildings to serve a number of different functions, like fuel production, diffuse daylighting, or food production, and really carbon storage. A lot of my work on the building scale is in collaboration with my colleagues at the University of Technology, Sydney. So I encourage everybody to really keep an eye on what they're doing because they have some really great large-scale installations going in Sydney that have to do with algae architecture. And the last thing that I did want to mention is going back, zooming back out into the biomaterials ecosystem. One thing that many of you may, or a frustration that many of you may share, is that there, as an architect, as an engineer, as a material scientist, like I definitely want more biomaterials to be available for use in buildings. But if we actually look at the current ecosystem of biomaterials and just taking agricultural residue as an example, we know that agriculture, distributed agriculture, growing straw or hemp or bamboo, even corn, even sugar waste like bagasse fiber, this distributed industrial ag has CO2 sequestration yields in the biomass every year. But that agricultural biomass residue is either burned, so there goes the CO2, tilled, or left to degrade. And in all of those scenarios, all of the carbon that was stored in the fiber is released back into the atmosphere. Unfortunately, there aren't very many industrial fiber processors in the States. After the Civil War, a lot of the fiber processing moved overseas. So there isn't really a link between industrial agriculture and industrial fiber. And so what that ends up resulting in is a lack of consistent supply of ag residue. And so the biomaterial manufacturers really struggle with sourcing the biomaterial. And a lot of times we have to look at other places like China and Europe to source a lot of the processed fiber. There are a few examples of some products that are popping up, but they are again more regional. They're a little bit more expensive. And so if you pay attention to kind of where the raw material resources are coming from manufacturers of hempcrete, for example, or other types of fiber-based materials, it's oftentimes coming from overseas. So what that leaves is architects and engineers hungry for carbon storing materials, but without the right supply chain in North America to have materials that are readily available and at cost parity. And this ecosystem is a little disjointed. I'm showing an example here of a company, and there are many companies that are actually proposing solutions to this in the form of trying to monetize the carbon storage in the form of carbon credits. And so a company, Aureus Earth, who I'm quite familiar with, is looking at, large-scale emitters have a problem, right? They need to offset their carbon footprint, and they're looking to offset it by purchasing carbon offsets, which could then be used to subsidize much of the supply chain, so that architects and engineers will eventually then have for their use carbon storing materials that are in fact at cost parity or even subsidized because of the large scale emitters who purchase the carbon credits that are subsequently then transformed into these carbon storing biomaterials. So I encourage those of you who are interested in being part of pilot projects, or at least committing to using carbon storing materials, to take a look at Aureus Earth and see what they're up to, because I think their perspective is a little unique in that they're focusing on actual physical storage and biomass versus other approaches like such as soil sequestration and agrarian practices. So finally, we've been on this journey today, but what I want to leave you with is really my vision for the future landscape of biomaterials. And I have this quote here that I share with my students quite often from Mary Shelley, where we really do think that we're pioneering a new way by leveraging the really remarkable abilities of biology to grow materials, effective materials, do it in a very resource effective and efficient way. One thing that I tell my students is that we just need to learn from history and not repeat it, so that we can actually wield the biotechnologies that we certainly produce, unlike Dr. Frankenstein with his invention. So really the future landscape in my eyes, if we fast forward a few years into the future, I really don't think that we'll be burning the limestone to make Portland cement. I don't think that we'll be mining ore to make steel or melting sand to make glasses for us. I really do think that we'll be turning to biology to grow the materials with which we choose to build. And I really think that's a wonderful world in which we can live. So with that, thank you for tuning in today. I'd be happy to take questions in the discussion part of this presentation.